Ugh, mutations are so hard. I don't even know what a mutation is. What exactly is a mutation?
Hi, I'm Beles the biology fairy, and I'm here to help you with all your biology needs. A mutation is a change in genetic material of a cell or a virus. A mutation can be big or small and affect lots of change or almost none. And mutations can occur at any point during DNA replication.
Thank you, biology fairy. Are there multiple types of mutation?
What a great question, child. Yes, there are many types of mutations. There are point mutations and base pair substitutions, missense mutations, and much more. There are nonsense mutations and insertion mutations, and I get too excited. Let's look at each one individually. Let's start with my favorite mutation, the point mutation.
A point mutation is a chemical change in one base pair of genes. This small change can lead to a complete change in production of a protein, leading to an abnormal protein. The point mutation can lead to a silent mutation, a missense, or a nonsense.
Wait. This is getting a little complicated. Can you go in depth on what exactly missense and nonsense is?
Of course! Let me display it to you. Silent mutation has no effect on the cell. Imagine if you were in the library and this person is a mutation. She is being silent in the library so she does not disrupt the studying of others. Missense is when there is still a code for amino acids, but it's not the right one. This is how sickle cell anemia is formed. That small change in amino acid leads to a complete change in structure. Isn't that crazy? Nonsense is when the mutation changes an amino acid codon into a stop codon, stopping the production of protein, leading to a non-functional protein.
Oh, that makes a lot more sense. You mentioned something earlier about insertion and deletion. Is that the same thing as point cell mutations?
Oh dear, you'll need more help than I thought.
As the name suggests, insertion and deletion happen when a nucleotide is either inserted or deleted. This leads to a frame shift. A frame shift mutation is when the amino acid chain is either shifted left or right. This has a huge effect on the protein. Insertion or deletion also can lead to a missense or nonsense.
Oh, that makes way more sense. So where exactly do mitosis and meiosis fit into?
Oh, Whinter, you have so much to learn, but so little time until your test begins. Mitosis is the separation of one cell into two identical daughter cells. There are two types of proteins that make sure that everything is going well. Those are called cyclons and cyclin-dependent kinases. The MMP, or maturation promoting factor, is a complex in the cell that lets the cell know that the cell should begin to enter M phase. Just like cancer cells don't pay attention to the signals that regulate the cell cycle, the person in the library doesn't listen as well.
So I know cancer is bad, that it doesn't listen, and then cancer cells can go on dividing infinitely if they are given nutrients. But what causes cancer? And what are proto-oncogenes?
There are a couple of ways cancer can come about. Cancer can be caused by mutations to genes that regulate growth and division. Tumor viruses can cause cancer, and oncogenes, cancer-causing genes, can also cause cancer. And for the proto-oncogenes, I'll let the narrator cover.
Proto-oncogenes are normal cellular genes that are responsible for normal cell growth. The proto-oncogenes can be converted into an oncogene by movement of DNA, amplification, point mutations. The conversion of proto-oncogenes can lead to a normal stimulation of the cell cycle.
The movement of DNA. If the DNA ends up near the active promoter, then the rate of transcription will increase. Amplification. The proto-oncogene increases the number of its genes. And point mutations, the gene expression increases in the proto-oncogene.
Apology, Beles. I read something earlier today that said something about RAS proto-oncogene and the P53 tumor suppressor genes. What are those?
Mutations in the RAS gene leads to a production of hyperactive RAS protein, and therefore cell division increases. This causes the mutations found in the DNA of one cell to increase to the DNA of many cells, spreading mutation. Now, mutations in the P53 gene prevent suppression of cell division, so the cell will divide over and over again without end. Both of these mutations increase the chance of cancer and are commonly found in those with cancer.
Multiple of these mutations are needed for serious cancer to exist. The instant that these mutations will occur increases with age. A cancerous cell normally has one active oncogene and the mutations of several tumor suppressor genes. It is also worth mentioning that you can inherit oncogenes or mutant alleles of tumor suppressing genes. The change of cancer can be hereditary. Inherited mutations in the tumor suppressor gene are common in those with colorectal cancer, and mutations in BRCA1 or BRCA2 genes are found in in 1/2 cases of breast cancer.
Divisions in mitosis lead to cancer. Oncogenes are cancer-causing genes, and mutations in RAS genes and P53 genes leads to higher chances of cancer and are needed for cancer to happen, right?
Exactly right. Cancer cells also lack density-dependent inhibition and anchorage dependence.
So cancer cells won't stop dividing even if there's not enough room?
That's exactly right. Now let's talk about the mutations in meiosis. The mutations in meiosis occur in anaphase 1 and 2. Non-disjunction can occur. A pair of homologous chromosomes do not move apart in meiosis 1, or sister chromatids do not separate in meiosis 2. Because of non-disjunction, one gamete receives 2 of the same types of chromosomes, and the other gamete receives no copy.
But what happens if a zygote is fertilized with a gamete that went through non-disjunction?
When this happens, we, in the biology fairy business, call it aneuploidy. This is when the zygote will have an abnormal number of chromosomes. A trisomic zygote has 3 copies of a particular chromosome. A trisomy on the 21st chromosome causes Down syndrome. The triploidy is an organism that has 3 sets of chromosomes. Did you know that bananas are triploid? Tetraploid is 4 sets of chromosomes. Polyploidy is a condition in which an organism has more than 2 complete sets of chromosomes. This happens more in plants, like wheat, who have 6 sets of chromosomes, and are more normal in appearance than aneuploids.
So the mutations in meiosis happen during anaphase 1 or 2 and are a result of non-disjunction. These mutations affect chromosomes, resulting in extra chromosomes or too few chromosomes. Some organisms, like wheat and bananas, have multiple sets of chromosomes.
Exactly right, Whinter. Now go get this test.